Thank you, everybody, for showing up this afternoon. I think we have a really awesome lineup this afternoon. It's going to be focused on feeding the future astronauts, but we have a lot of diversity in the presentation, so it's not all exclusively about feeding the astronauts and the future colonists. It's going to cover a range of things. My name is Ron Turner. I'm from a not-for-profit company called Answer Analytic Services. If you were an early register for this conference, you probably saw the name Penny Boston instead as moderator. Unfortunately, she got called away, and they needed someone to come and herd the cats today, and they thought that I was a good cat herder or squirrel herder, and so they nominated me for the task. I have since discovered that I have some reasonably well-behaved panelists, and I may not have to call upon those skills. Without too much further ado, I'm actually going to introduce the speakers as they start their talk rather than going all the way down the line. You can certainly see their names on the screen behind me. But the first speaker is Lynn Rothschild from NASA Ames.
Thank you very much.
And that's all I need to do.
Okay. No, I'm not finless. There. That got moved around. All right, very good. Let's go from there. I'm not used to sitting down when I speak, so that's going to be the first thing I'm going to make the herders allow me to stand up. There we go.
All right,
okay.
so I was wrong. I
Now
have to herd some cats here.
Of course. Chief Cat here. I don't know if you can bring the lights down a little bit or if that's a problem with the filming, but if you can, I'd like to start by saying that I actually wear two hats at NASA. When I got to the agency several decades ago, I became, fascinated by astrobiology. I'm an evolutionary biologist and microbiologist by training and by love. And so it's been a privilege for many years to get paid to look for Martians and Europeans and so on and so forth. And there I am up in the Bolivian Andes. But about 12 years ago or so, I was asked to start a program in synthetic biology for the agency. And I sort of laugh a little bit because I've been in some talks with Chris Carberry over about 3 years about this, and I think it's taken him a while to get the idea that you could actually have biologists at Humans to Mars. So this is my first Humans to Mars, and I'm very grateful to be here. So what I'm going to do is tell you a little bit about the SynBio shtick and why that is so important for bringing humans forth off this planet. So when you're on planet Earth, you have all sorts of needs as a human. You need transportation. You certainly need life support, food, oxygen, medicine, waste, recycling, clothing, and so on. People in the military tell me they've got just as hard, if not harder, problems going to Afghanistan. And I say, yeah, but you get oxygen and gravity, and we don't even get that. So we clearly have the harder task. We need habitats, we need power, heat, light, radiation protection, and certainly communication. But we have a lot of challenges when we go forth from the Earth, and the big one is upmass, which you don't have the problem on the Earth as much. So someone has a piece of equipment in a lab that's 300 kilograms. I want one that's about 3 grams. So that is intimately tied in with the cost to go forth. We have to worry about storage, we have to worry about reliability and flexibility, and we have to worry about power. Though I say Amazon.com just does not have overnight delivery to the moon or Mars and is not likely to anytime soon. So this is the ultimate field trip. You better have what you need with you. So what's the solution for this? And I think that we've been hearing a lot of solutions over the last day and a half, the sort of the big boy, bring up the steel sort of solutions. But what I'm going to do is pitch the idea that the solution for this is life itself. And so what I want you to do is to think of life not as something you're bringing forward or something that you're looking for, but life itself as an enabling technology. And in addition to that, we can tweak life with something called synthetic biology, which is basically the design and construction of new systems that are not naturally found in nature. This is the British government's definition. We've pretty much taken this in the US government. I've been representing NASA and White House working groups in synthetic biology. And if that's too many words, that's probably as good a definition as any. So what's the big idea about this? And that's what I'll take the rest of my time, is giving you a few of the many, many ideas that we've been putting forward and that many other people, I'm sure, are going to be starting to think about in the next few years and go forward. So, for example, we use organisms for material production on the Earth. Virtually everyone in this room, just quickly looking, is probably wearing something that was made by an organism, whether it's cotton or wool or silk or leather in your shoes, and so on. But we're not going to take a lot of these organisms with us. We're not taking sheep and we're not taking trees and so on, even though these are the sort of organisms that produce most of our biomaterials today. So we would love to have some of these materials, the cotton, the spider silk, which is stronger than steel in cross-sectional area, cotton, and so on. But we are going to take something like this. This is Bacillus subtilis, which is a bacterium that produces incredibly resistant spores. Those white dots are spores. Bacillus subtilis has been flown exposed to space environment for nearly 6 years on LDEF. So it's already proven itself to be extremely space hardy. So why not take something like this that's already proven itself and engineer it using the tools of synthetic biology so that it makes latex, it makes cotton, and so on. In fact, I don't know if Jerry Sanders is still in the room. I'm pretty sure he was sitting next to me at a meeting years ago when we were starting to pitch this. And he said, "You think organisms are so smart," and it may have been someone else, so I may be being a little unfair here. He said, "Could you ever engineer an organism to make rubber?" It's like, rubber literally grows on trees. You do know that. So yes, biology can do all these things. So what can you do once you make materials? Obviously, you can make clothes and so on, but you can also make habitats. This is fine if you're going to the moon for 24 hours or so and you're coming home to ticker tape parades. You wouldn't want to do this for months and months on Mars or a human colony on the moon. You might want to do something like this, and this is an image courtesy of our colleague Chris Maurer, who is an architect in Cleveland. This was a project funded by, still is being funded by the NIAC program, and our idea is to use fungi actually to build this structure. So you don't want to actually take the mushrooms. It's not like we've stuffed mushrooms in a plastic bag, but the underground part of a mushroom is called a mycelium. So these are these hair-like structures that form underground. And there are several companies that have discovered that if you take a mold and fill it with the mycelial bits and something for them to eat, and what we're talking about is literally garbage. Lawn scraps, we fed them coffee grounds, they get a little caffeinated after a week, but they literally can eat garbage. You can turn into these material things. Now here's some of our mycelia growing on Martian Regulus simulant. Happy to do that with a little extra nutrients. Here's a stool that my students, oops, I don't know why the video didn't work, but there we go. One of my students made last summer. It's been baked, and you can see it's rated for the average Stanford student. So it's pretty quick and simple. So imagine you have a double plastic bag with some mycelia. You add a little growth medium, and it grows to fill this, and you've got now a structure that's growing in place so it can be sensitive to the environment. You can make last-minute changes. Oh, dear, I'd like to have the, whatever there. You can do it. You're not wedded to these steel structures that have been sent up. Another big idea, we can send digital information to reprogram organisms. So at this point, we write code for DNA in the lab. The students hit send. We, at a different computer, hit send with a credit card number, and presto changeo, some DNA synthesis company makes the DNA. So you've broken this physical link now, and it doesn't matter whether you're talking about across town or across the solar system. You've broken the physical link between the information in DNA and the actual physical DNA. So biology does chemistry on the Earth. Why shouldn't it continue to do chemistry for us on space? We've been harnessing the power of chemistry for thousands of years, and that's probably as good an explanation as any. Nanotechnology, we clearly need nanotechnology moving forth beyond planet Earth. We're not going to take massive cold rooms and so on, but guess what? It just so happens that we are incredibly fine nanotechnologists, we meaning life. And think about it, we use polymers like DNA and proteins and so on in our cells. Every last monomer better be in the right place or you're sunk. That's something that is beyond the capabilities of any nanotechnologist I know of, no matter how many awards they have. And so one of the great projects that's been going on in my lab with an NSTRF fellow funded by Massa STMD, my sponsor's in here, right here to my right, so I have to be very careful, is using DNA itself as a wire. So you can take it, you can program it so that you put certain nucleotides in the right places, and you can incorporate silver atoms in exactly the right places and make an electrified wire. Cheap, easy, no cold room needed. You can actually pull metals out of things like integrated circuits, because there are a lot of metals that we normally bind in our bodies, and all organisms do. I always tell people, look at the back of a vitamin bottle. There's the back of a vitamin bottle if you want sort of a quick list of some of the easy ones. And so we had a project funded through NIAC to take some of these bits of proteins that bind specific metals, to grind up an integrated circuit, pull them out, and then be able to reprint a circuit. And we've done a proof of concept with copper on that. We can use chemical energy to make electrical energy. It's not just electric eels that do that. Every person in the room is taking the food they ate at lunch and using it to keep your brain functioning. We could make life DNA 2.0, where we have organisms with right-handed amino acids and left-handed sugars, opposite of what we have today. And we can take some of these capabilities, like the ability to live in extreme environments, like here's a hot spring in Yellowstone, and push organisms so that they can live in more radiation-enriched environments, or higher salinity, or colder temperature, or what. It's a project my students call the Hell Cell, which I love. Okay, so you can do all these things. And now it's time to take some of these ideas to space. And so I'm very proud to say that I'm the PI on a secondary payload on a DLR satellite, the Eucropus mission, which finally launched in December 2018. There was much rejoicing. The beauty of this is that we're testing some of these basic techniques in synthetic biology at microgravity, which we've now completed, lunar gravity, which we're in the middle of, and we will then be able to do Martian gravity when the spacecraft spins up in a few months. So the final thought is that we've got all these needs for human settlement, but these constraints that we have that my colleagues who do ground-based synthetic biology don't have, I believe is going to unleash creativity that's going to allow us to make ground-breaking and game-changing solutions, not just for space exploration, but for humans back on Earth as well. Thank you.
Thank you, Lynn. We're going to hold the questions for a broader general discussion at the end, as it's for these panels. Next up is going to be Mike Selden from Finless Foods, and he's going to be talking to us about clean meat.
I am. I'm going to keep a timer so I can actually make sure that I'm doing this right. Cool. So it works. So what I'm going to talk about is food production, and a lot of the work that we do is geared for food production on Earth. That said, it's extremely applicable to food production in space, and there are people who are doing what we do that are like extremely focused on that specifically. So what we make is fish, and when people think about the way that fish is made, they're generally thinking about, boats, nets, the ocean. But I am a cellular biologist, and when I think about how fish is made, I don't think about boats. I think about cells. Satellite cells exist in the muscle tissue of any given animal, and these things divide and divide. They create more of themselves and sort of wait to be used. They end up being used. They differentiate into separate terminal lineages. So basically what that means is they end up as a few different types of cell. There's myocytes, muscle cells. These are what give you the protein that you're interested in when you're eating, and they sort of have a bunch of cellular function as well. Let's talk about food instead. So there's myocytes, there's adipocytes, which are fat cells. They provide both most of the taste that you get in fish, as well as a lot of the nutrition. Those long-chain fatty acids, DHA, omega-3, omega-6. And then the third type that is most common in muscle would be the fibroblasts. So these are the structural cells that secrete ECM. So they basically create the mouthfeel. They create the texture of the food that you are eating. So these three cell types will grow out, bind together, and form these muscle fibers that end up as meat. And so at Finless Foods, what we're doing is we are basically redesigning meat. We're going back to the very basics, and we're forgetting these sort of intermediary steps of boats and nets and the ocean. We're growing fish directly from cells, the way it's grown inside of a fish, outside of a fish. So it's not a vegan or a vegetarian replacement. It is not the Impossible Burger. It's not made of plants. It's made of real animal cells. I have to repeat that every time, else people get confused. So what we're doing is we're isolating cells from a real fish. And theoretically, there's only one sample, for us, the size of a grain of rice, needed from each species of fish once. And you're isolating these cells of interest. You're then cultivating them in a feed, right? You can't make something from nothing, so you have to feed them basically what they want in order to survive, and then you give them a structure to give it the same look and feel of meat. So these 3 cell types that I talked about, the muscle, the fat, and the connective tissue, they are what provide the experience of eating meat. But there is one really important thing that they can't do once they're fully differentiated, which is that they can't divide and create more of themselves. So what we're isolating is a cell type that can do 2 things. They need to be able to divide and create more of themselves, so we can actually expand the population of cells that grow the meat. And then 2, they need to be able to differentiate. They need to be able to turn into these muscle, fat, and connective tissue components that can end up becoming all the function of the meat that you actually want to eat, rather than just raw biomass. These cells eat very simple things. They eat salts, sugars, and proteins. And this is, I think, where I can start injecting space into this talk, which is not something I normally talk about a lot. But basically what this does is it gives us the opportunity to take ingredients that are considerably easier to grow from a small source. You can do salts and sugars from like plants, essentially, and distilling salt, and then the sugars from plants, and then the proteins. These are things that you can create using microbial fermentation. So like using things like yeast, which are really easy to ship up to space in really tiny containers. So basically, the salts and sugars are food safe, food grade, and then the proteins are something that we're producing in-house using fermentation. So we're brewing them like beer. And so what this means is in order to have fish meat in space, you have essentially a few options. You can either make it here and send it up, which is not sustainable, right? That's not actually creating a self-sufficient human life-supporting system on the moon or on Mars. Or you can find a way to ship an entire tuna living into space. You'll need a few of them. You'll also have to figure out how to get them to breed and create more of each other, which we're having trouble with on Earth as is. And in case people aren't clear on that, a tuna is about my size. I'm very large, and you don't want to send lots of me into space. So basically, this allows us to actually feed people in space meat, but using resources that come from plants and come from things like yeast. So for us, since we're growing cells, we can actually really get rid of a lot of the variables that make fish farming complicated. Fish are all different from each other. They require very different amounts of space. They're different sizes. They require extremely different temperatures and grow environments. But for us, a cell is a cell. And so for us, culturing a cell from the most expensive fish you can possibly think of, for example, bluefin tuna, which is what we focus on, or culturing a cell from the cheapest fish you can possibly think of, like tilapia or carp, it's really the same for us. So we say, why not just start with the high quality stuff? Why not just start from the top and work our way down? And basically, make our process more and more efficient until eventually we can work on commodity good fish. But I guess astronauts can't be too picky, right? They'll eat what we send them, apparently. So anyway, there are a lot of advantages to this, and this sort of comparison is for fish farming on Earth. The most efficient system that exists on Earth today is salmon mariculture, which is salmon that are grown in these nets. They're about 50 meters in diameter, mostly off the coast of Norway. So what we can do is basically condense that. So we can do about the same amount of production as one of these 50-meter in diameter nets with these two 5-meter in diameter footprint total, these two bioreactors. So basically, we can produce way more food with way less space. And you don't have to rely on the ecosystem like the ocean. This salmon system is only so incredibly efficient because the fish are able to eat a lot of the things that are just floating free in the water. We're not going to have that luxury on Mars or the moon, at least soon anyway. And so basically, this creates a controlled, sterile system that can do much better efficiency. And so the one thing I actually want to point out as well, is like, this is the most efficient fish farming system that exists. This is salmon. But this is basically middling pharmaceutical numbers for cell biology. This can be bluefin tuna and still beat the salmon. This can be whatever type of fish you're interested in. And one of the reasons that this gives us this efficiency is because when you're farming fish, you're farming a lot of stuff you don't really need. There's bones, there's skin, which some people like, but there's blood, there's organs. And the fish themselves burn energy via moving around. They have heartbeats, they have other organs, they you're putting energy into a system and then it's being burnt, creating all this stuff that you actually don't need. And so we think that you can create a much more efficient system by narrowing your focus and just making all of your inputs go directly towards the desired outputs, which is the fish meat. So yeah, these are numbers for salmon. Only 56% of it is edible fish meat. The rest of it is byproduct or gets thrown away. So basically, the media is excited about this, mostly because of things on Earth. People really see that there's a lot of problems with the food supply as it is today, and I know this is like injecting Earth agriculture into this, but I think it's important. I think most people are kind of cognizant of the fact that fish now is like really heavy with like mercury, with plastic. People are using antibiotics, they're using growth hormones, and we're destroying our ecosystem. And so this is basically a way to circumvent that, create something that is healthier, that is more efficient, but still is the food that people really, really love without forcing people to be vegan. And so one of the other things that's cool about this is we can actually create extremely high levels of control over what nutrients are inside of the fish. Are there certain nutrients that are really important to the diet of specific people, say people in space? I'm not an expert, you all probably know better. But basically we can actually make sure not only that it's the exact cut that people want every single time. If people want tuna belly, we can just produce that and nothing else. But also we can actually increase levels of nutrition because we have increased levels of control over the system itself. So I want to take this out of the realm of like sci-fi though and just show that like this is a thing that is real and exists. So what I'll show you right now, I hope if it turns on, is a video where we had our first prototyping. So we got a reporter from The Guardian to come in and taste some of the fish that we had made in our facility in San Francisco. So basically we gathered 25 of our friends together and had for the first time fish that was entirely grown outside the body of an animal. And whoever's controlling this might have to click it. Anywhere. Wrong. That's my fault. Sorry, I'll stop pressing it. If you go down to the bottom, there might be a play thing there. If not, I'll just have to leave it and people can find it on the website.
No, I'm not seeing anything.
It's okay. So anyway, if you want, you can find us online. We're Finless Foods. We definitely have that video online, along with a bunch of others. But basically what we're attempting to do is take these like cells, these initial isolations of cells from real fish, and use them to change the entire way that seafood is made from the ground up. I'm Mike Selden, co-founder and CEO, and I'm looking forward to the discussion on the panel.
Okay, that was awesome. Thank you very much. Your mic can go off. Next up is Anjan Contractor he's the CEO and co-founder of BeeHex, and he's going to tell us a little bit about BeeHex and intrigue us with some of his ideas.
Thank you. Thank you for giving me an opportunity to speak at Human to Mars conference. In fact, we presented our very initial work in 2013, and I want to talk about what has been done since then. So I'm a CEO of BeeHex, and we manufacture 3D food printers. And today my focus will be how 3D food printing can be beneficial to human space missions, especially Mars. Going to Mars is a very challenging task. One of the major challenges is what sort of food and nutrition astronauts would be consuming. Just imagine going to Mars taking 5 years. Now, during 5-year time period, what can happen to human body? What can happen to human physiological health and mental health? And food is very essential to that. Now, if you think about what astronauts currently consume at the International Space Station, it's meals ready to eat or MREs. They are prepackaged food, dehydrated, very boring, very limited choices of the menu, and it creates a lot of waste. Not only that, think about my earlier point. When astronauts spend 5 years in space, what are the chances they fall sick? If they are more than 50%, then they are more than enough. Will they have access to nutritionist or dietitian? If they are falling sick, will they have access to medical equipments, medical specialist? So food, if we combine with nutrition, can provide a lot of benefit. And that was the premise of my earlier work in 2013 with Johnson Space Center with Advanced Food Lab. And in 2012, there was a call from Johnson Space Center about microencapsulating micro and macronutrients so that they can extend the shelf life. And the idea was that this sort of nutrients astronauts later, when they go for deep space missions, can use as a source of food, or partly source of food. And this work is going on for quite a few years, in fact, quite a few decades at NASA, about dehydrating food, taking the moisture out of the food system, and encapsulating it so it can have a longer shelf life. But will it have a shelf life of more than 5 years was a question, so that was the research topic. My background is in robotics and 3D printing, and I have worked on quite a few complex composites, plastics and composites for NASA. But this was something that I looked at, and our team thought NASA does not want dehydrated food that they can rehydrate and drink, or something that doesn't satisfy their appetite. Also, we came across quite a few research papers which talked about chewing, releasing endorphin in the body, meaning your mental health is directly related to chewing. So chewing is very important. So what we proposed was, okay, use the dehydrated powdered ingredient, microencapsulate that so you can have a longer shelf life. Not only that, but contain it such a way that they are in very inert environment, and then transport those food, micro and macronutrients, building blocks of food, into a mixing chamber where we introduce water and oils and make it in a viscous form, and then transport them into a robotic system such as 3D printer that can recreate texture. So this project was funded in 2012-2013, and in phase 1, we 3D printed pizza, and that was my earlier work in 2013 that I represented at Human to Mars conference. Why pizza? Because pizza is very natural to 3D printing process. Most of you know by now what is 3D printing process. It's layer by layer. So we were trying to find out foods which are layer by layer. And pizza is very natural, dough, sauce, cheese, toppings. And one of the challenge of this project was to start from dehydrated ingredients. So everything that we started out was from dehydrated ingredients. We rehydrated them. The rehydration chamber is in fact a separate system. And then the same system, the rehydration system, creates cartridges. We take the cartridges, plug it into the system, into the prototype, and we tune astronauts' physiological health and needs and taste, and it will create pizza. So it can create a small pizza, snack bite pizza, or it can create very large pizza. And it can also tune what sort of vitamins are going into it. So out of that work, I decided, why don't I commercialize this technology? So we decided to spin off a company, and we were published in a 2019 NASA spin-off. That's how BeeHex was started. So what we are doing right now is essentially the same. What we are providing to astronauts is what we want to provide to the rest of the world. Now think about what is personalization. Right now, personalization to most of the people is based on taste preferences, or gluten-free, or low fat, or low sugar, or most recently you have personalized meal plans. But what if you have a machine that can take into account your genetics, your physiological health, your vital health, how much sleep you have had last night, and based on that it tunes your diet. So we first started building algorithm, the back-end algorithm, where you can plug in, if you have 23andMe data or Ancestry.com data, raw genotype data, we can use that on our algorithm. If you have a ubiome or your gut biome, your metabolic health, essentially the same thing. If you have that data, you can plug it into our system. Your blood marker, same thing. If you do annual checkup and blood testing is what you usually do, we can take that data into account. Your vital signals is something we can read from your Fitbit or Apple Watch or Nike Watch, any platform. If you have allergies, you can input that manually. Your physical attributes, your weight and your ethnicity, and et cetera, your age and health conditions. And then what we generate is a machine code or a G code that talks with our hardware. And this is the hardware that we have in place today. It can take about 12 nutrients in the paste form. So after we have cartridges coming from the rehydration chamber, we can plug into this machine. In the center, we have 24 granular or powder form ingredients. Once the information, your genotype information and all those information plugs into this hardware, it can start creating whatever you want. So for example, if you want snack bars, you can create snack bars. Over here, this is on the left side is the first snack bar we created at our company based on my physiological data. What I needed was proteins, many different types of proteins, many different types of fibers and carbohydrates. And I tuned my taste for chocolate. So you can see the chocolate bar being printed. After printing, we thawed it with a liquid nitrogen. It made it hard, so I could take it out and eat it. One on the right is my co-founder. My co-founder required probiotics, protein, and carbohydrates. So you can see the 3 layers built up, which didn't require post-processing. So that's the type of technology we are developing at BeeHex. But we always think, what is next? One of the things we have started investigating is in vitro tissue growth. Using the same platform, same 3D food printing platform, but having different software, we were able to recreate cardiac tissue patches. And the one on the right side you see is a cardiac tissue patch. We started out with a CAD model, and it's essentially the size of the penny, about 19 centimeter in size, diameter in size. We first created halogens, then we, in the cavity, we 3D printed alginate and the cell mixture and the nutrient mixture with a tuned pH. And then we deposited calcium chloride, so it became solid medium. And then the same in situ we started incubating. After incubation, we had a pretty good yield of the cardiac cells. So this technology is something that will change the whole perspective of 3D food printing. Mike talked about the 3D meat printing, which we haven't investigated, but that could be done with the same platform. So that's in a nutshell what BeeHex is up to. Thank you very much.
And closing out the panel session, Gioia Massa from NASA Kennedy Space Center.
Okay, well, thank you, and thank you to the organizers for inviting me to be here today. So I'm going to go to something maybe a little more prosaic, which is plant food crop production. And I want to talk about the work that we're doing at Kennedy Space Center. We've actually been working in this area for more than 30 years at KSC, and it's evolved over time. So I'll give you a picture for that. This work is under both NASA's Space Biology program as part of NASA's Space Life and Physical Science Research and Applications Division, say that 10 times fast, and the Human Research Program. So what our goals right now with the food production system are to help ensure food system security on long duration missions beyond low Earth orbit. And so what I mean by the food security is to have continuous access to sufficient, safe, and nutritious food, which helps meet the dietary needs of the crew, but also enhances, their health and their performance. And proper nutrition is going to be really critical. And one of the challenges that we have with the current space food system, which is somewhat limiting, as Anjan talked about, we actually have over 200 foods in the packaged diet, so it's pretty good, it's pretty diverse, but over time those foods degrade, both in terms of quality and in terms of the vitamins in those foods. So that right now is probably our biggest risk going forward for Mars. We also think that we can help provide safe, nutritious, acceptable fresh food and add variety to that diet. And food is important to morale. I mean, food is a really important part of almost every culture on Earth that I'm aware of, and it's a place where people come together. It's just a really important part of everyone's life. So it's probably going to be equally important, if not more important, in space where you're limited by your surroundings. So in the near term, we're looking at nutrient supplementation of the prepackaged food system. We think that adding fresh produce can supplement some of these key vitamins that break down or nutrients that may be low in the packaged diet. You're also adding this bioactive compound. So it's not just take a vitamin pill, but you're adding other sort of bioactive compounds in the fresh produce that have health benefits that we're only discovering, all the time. We are looking at this point at pick and eat crops that you can pick and eat fresh that don't require a lot of processing and preparation. We don't have any way to cook anything on space station. We don't even have an oven or a microwave. We're not going to probably have that on Gateway or on a transit mission because space is so limited. So we need to look at foods at this point that you can eat with minimal preparation. And these fresh foods can add variety to this packaged diet, allow some customization. You can make lobster salad wraps with your prepackaged lobster salad out of your lettuce, for instance, and the astronauts on space station have already done that. And also allow some, psychological appeal. And one of the things we're trying to get a handle on is how do growing plants, how do tending plants impact the crew behavioral health? So we're actually starting, studies on orbit now where crew help us to understand, not just anecdotal evidence, we loved growing the plants, but what is growing the plants actually doing to behavioral health and psychological aspects of living and working in this extreme environment of space. But we also have to maintain food security and make sure that astronaut health is kept as the primary priority. So food safety becomes a real issue, and looking at the entrance of things like pathogens to the system, making sure that we have all the hazards mitigated as much as is possible. So we want to be able to test any of these space food production systems before we actually become reliant on them. And that's why the International Space Station is such a good test bed. We're getting fresh prepackaged food up there all the time, as well as fresh produce that gets sent up. That's, a very limited life item. So they're not dependent on supplemental food production, and we can test it in kind of a safe environment at this point and get it working really well before they're actually relying on that as a source of their vitamins. And then we're limited, of course, by the vehicle limitations. In the long term, we want to do more caloric substitution with actually growing crops to get to the point where we are self-sustainable for life off Earth. And there's a lot of challenges to get there, but we'll be looking at those staple crops, and we've done a lot of work on those over the years to get towards this bioregenerative capability for sustaining the crew. So here are some of the ideas of candidate plants. Right now, as I said, we're looking at kind of the salad crops and the herbs, things that you can add to that packaged diet. In the longer term, we have these staple crops, and we think of things like sweet potato and white potato as kind of an intermediate crop because they require pretty minimal preparation if you have something like a microwave or an oven to cook them. But things like wheat or soybeans actually require a fair amount of food processing equipment. You have to do your milling and your grinding and your, your tofu pressing and all these things. That's mass that you would have to launch. So that probably wouldn't be till farther down the road until we have more of a longer term research station or a permanent presence on a surface system. In terms of the challenges, we have some that we think of as kind of more of space, challenges, your microgravity and fluids issues, which are actually one of our #1 challenges on the space station. We have surface challenges like your dust, your micrometeorites, some shared challenges, things like radiation. We have crop challenges in terms of, how productive are the crops? How tolerant are they to stress? How do you optimize the environment for those crops? And then we have challenges that share all of these different scenarios. How to keep your plants small, how to have the food safety managed, and how to be sustainable with your growth system. So there's a lot of questions. We actually are recruiting a lot of help from universities, from middle schools, high schools, and companies and partners from around the world to help solve some of these problems. Here's an eye chart, so don't read it, but you get a feel that we're working from our ground research and space station research towards the gateway, lunar surface, Mars transit, and Martian surface. And so we have challenges both of radiation and gravity, and these differ in the different environments, and we're trying to build upon those and figure out the key risks. So this is kind of our strategic plan and how we're going to get there. So you can see a lot to do. And right now, what we're doing is we're working on the International Space Station. So this is our Veggie hardware on the International Space Station. Veggie is our vegetable production system, and you can see astronaut Scott Tingle. We actually have 2 veggies up there now, and he was growing simultaneous in both Veggie units, doing some mixed cropping in there. Veggie is a very simple system. It's a low mass, low power, low volume system because it's collapsible, but it actually is fairly crew time intensive. And we're learning a lot about what to do and what not to do fresh produce production in Veggie. We're also developing a hazard analysis for the food safety, monitoring microbes, monitoring how the crops grow. One of the second Veggie experiment that we did, we actually had the harvest live on NASA TV, and the astronauts who did the harvest made some speeches, and I really like the points from their speeches because they really encapsulate all of what we're thinking about for crop production in space. So Scott Kelly was talking about the logistical complexity of living and working in space for long periods of time and the long supply chain that's needed, and how for Mars we have to become more self-sustainable with our food supply. And Kjell Lindgren, who's a physician, had a slightly different take. He talked about the benefits of eating fresh food and the contribution that the plants had to that small ecosystem on the International Space Station, as well as the psychological benefits of seeing green growing things up in that environment. And, I think the psychological benefit is something we can almost not speak highly enough of, just because when you're so far away from Earth, and if you're on one of these very long duration missions that we've been talking about here, you're not going to see the planet for a long time. And I think that having a garden, having that little piece of Earth with you could be really, really important. Could also be really stressful. What if your plants die? How do you handle that? So we want to get a handle on all of the different repercussions of this, but we've heard astronauts talk about how plants help mark the passage of time. When you're going around the Earth and you have a sunrise and sunset every 90 minutes, it's kind of easy to lose track of time. When you're on your way to Mars, you don't have a sunrise and sunset. It could be even harder to keep track of time. But watching that tomato grow and ripen and have that sense of anticipation that you're going to harvest it soon could be really awesome to keying you in on that. The other large plant chamber that we have on the International Space Station is our advanced plant habitat. This is kind of the opposite end of the spectrum from Veggie. This is a highly controlled environment. It has over 180 sensors and control systems to kind of control and monitor every aspect of plant growth. And APH is something that can be almost entirely operated from the ground. The astronauts kind of put a tray in, turn it on, and then ground controllers do the rest. It's a really high quality plant physiology instrument. And so Veggie's more of a garden, and we're learning lessons from both of these units that are helping to drive towards what we come to a future food production system. So we're really trying to figure out, okay, what complexity do we need? What automation do we need? There are a lot of aspects of this where autonomous operations or robotics might be really important. We don't want to send people to Mars to spend all their time doing subsistence farming. You know, we want people on Mars exploring Mars. So we are going to need those aspects. Some of the other things that we heard about today, I think, are also going to be really important in future systems. You know, we're looking at perhaps genetically engineering custom space crops that are going to really meet the needs that we have while growing well in our environment. So there's a lot of forward work in this area. And as we think about what, you know, a growing system might look like on another surface, you know, we can think, well, in a more near-term Mars landing where we have this sort of research outpost, maybe we'll have something where we have a large chamber for fresh food production. You can see this would be something that would launch on a rocket. And it looks remarkably like the garden or the growth chamber at the South Pole Station, the U.S. Antarctic Station, where the crew that overwinter in Antarctica spend a lot of time and they get fresh produce. So I think that that's going to be important. Maybe we could have deployable surface systems that could either be pre-deployed or they could be sent out from a habitat. They could be banking oxygen, you know, one of the nice little byproducts that plants are generating. They could be pulling CO2 out of the Martian atmosphere, and they could be storing produce for when astronauts arrive. So, thank you.
So we're about to segue into the question and answer period. Let's give one more round of applause for our speakers. And is there anyone poised to ask questions? All right, let's use the mic, and looks like we've got several lined up.
Just listening to this, going from end to end of the table, one might get the impression that there's a disconnect between the state of technology that NASA's looking at with, you know, essentially farming, and the state of technology that is really coming out of the lab. I'm just wondering, is anybody really looking at the trade-offs between, I guess, maybe the infrastructure that you need to grow these lab-based things that might look identical to plants or might look or taste identical to plants or animals versus actually growing the plants and animals?
Well, I'll start, and you know, I'm sure Lynn and others have comments, but you know, everything is a trade-off, and there are so many trade studies that we have to do. You know, even from the plant side, if we're talking a surface system, we need to do trade studies between looking at in situ resource utilization and trying to do everything in a recycled hydroponic system, or, you know, so. Yeah, I think we are considering the trade-offs. Some of the technology is at kind of less mature levels than others, so it makes it a little more complicated. And I know that we've worked hard and we're continue to work hard to kind of develop metrics to even be able to compare these things, because you're comparing, you know, vastly different systems. And so how do you roll those up into kind of a good comparison? I mean, they may have different mass, they may have different power usage, different volume, different crew time implications. And so how do you capture all that? So there has been a lot of work at NASA over the years in, you know, kind of the area of bioregenerative life support to capture things in a metric called equivalent systems mass, where you can use equivalency factors and kind of roll up mass
mass volume, power, cooling, crew time, and mission duration into kind of one number, but that doesn't account for things like the psychological impact or, the quality of the crew, experience. So we have to come up with really good metrics before we can even really understand some of the trades. But yeah, there's definitely trades.
The chair's prerogative, can I jump in? And do you have, for example, design reference mission mass for a 1-year mission if it's strictly packaged food, counting the packaging and everything? Are there mass fractions or mass numbers that are, to be compared with, that you can quote?
I can't quote them because I'm not the food person. But yes, we have that information, and those are the standards that we look at. And there have been studies, and Anjan, you were referring to them, about looking at the prepackaged food versus taking bulk ingredients versus trying to grow. Say you're doing wheat to make flour, and then you need your bread machine and your pasta maker, and so they do. Mission duration is kind of where things trade. So you can look at the graphs of all these and see, okay, the packaged food has a lot of packaging material, a lot of waste there. Is it a waste that could be turned into another resource, like a building material, or something to help stabilize the regolith? So that might be a way to help reduce some of these impacts. But yeah, over a certain period of time, certain things trade better than others, but I don't have the numbers on my head right now.
25,000 pounds over for a Mars return mission for
25,000.
Just shy of 25,000.
And is there a volume? Is it mass or volume limited? That was massive.
Yeah, and for us, comparing like what we're making, which we call cell-based tuna versus current animal-based tuna, the comparison is a little bit harder and a little bit easier. It is easier because currently tuna really can't be farmed, which also makes it harder because I don't have numbers on it because it's currently unsuccessful. And if we ever were to attempt to have a lot of these different like marine systems in space, I mean, you would need to ship out or distill and to keep track of large tanks of water or terraform these places to have oceans and lakes. And I don't know how hard that would be, but it sounds really difficult.
So speaking to that level of difficulty, can you repeat the term that you used, equivalent systems, or?
Yeah, it's called ESM, or equivalent systems mass.
Okay, equivalent systems mass. Mm-hmm. I really like this, and I really enjoyed it because it adds the next layer of complexity on top of the ISRU discussion around the scaling of plants to integrate, the biological systems as well, and the materials that we'll need to use and those additional products. How do you see yourselves integrating, because each of you relies on a substrate or feedstock material and generates some forms of waste in the course of producing whatever is produced according to whatever system. How do you see yourselves integrating those with the waste streams or with the other available materials that come online and become available as the other ISRU systems scale?
Okay, well, I'll start because I think that's fascinating, and I don't think it's an all or nothing. Like, we're not going to just go, oh, we're only going this route. I think anything we do is going to be kind of a hybridization. So, the things that we're talking about here are not mutually exclusive and should never be considered that. They should all be considered parts of this integrated system. I think we need to stop thinking about waste and start thinking about resource and figure out how we can recover as many of those resources as possible. And with biology, you can do a lot of that. And the things that you can't recover with biology, you can maybe recover with chemistry or find a different use for them. But we can't just be hauling all the fertilizer to grow plants in space, so we have to figure out how to break down inedible materials of plant waste and food process waste and human waste and retrieve all of those resources. I mean, even you look at our planet today, and we have these landfills everywhere, and it's like these are gold mines, and we should be approaching them as such. So I think we have to definitely do that better in space.
Well, Ruth Hammersbach from the German Aerospace Center in Germany. I would. Okay.
Well, I wanted to just make a comment to that. I think that's a great point. There is a lot of hypothesis than a real work at this point. What we have seen by far, or what we have looked into by far, is when we want to reintroduce microbiome into food, they are mainly in your gut. So we want to recycle the microbiome and put it back. Now it sounds gross, but it is something that is very practical. Same can be done with a lot of proteins. You have to do quite a few chemical synthesis in the different subsequent processes, but that is something that a lot of researchers have recently started looking into how to do that.
Yes, okay. So I appreciated your talks, and I would like also to stress on waste management. And Lynn spoke about the Eucropis mission, that the main idea of Eucropis, it's in the name, it's euglena and a combined organic food production, meaning that there is a filter system with biology, with microorganisms, and we put in urine and we degrade the urine to get nitrification, for plant growth and water back. And so I think this is the main goal, to take the waste and bring it back to food. And so I would like to add this point also. And for the talk of Mike, for cell culturing in microgravity or under partial gravity conditions, did you test on ground? There are some facilities available, at least to get some idea how cells will behave under altered gravity condition, how you culture them there. Do you have approaches like this?
No. I'll admit, our
so
company
we can
was really
talk later. I have some ideas.
I'm definitely interested. So our company was very much formed to solve like environmental problems that we see on Earth. I think that there's a lot of like synergy between us and space food production, and I'm super interested and would love to learn more.
So I'm responsible at the German Aerospace Center for the ground-based facilities, and a cell biologist. So it would be nice to talk later.
Absolutely.
Okay.
On that point, there is some work done on yeast in microgravity. So you want to take a look into that. But the conclusion of the research work that I have come across of yeast is that you can have healthy yeast growth compared to Earth gravity in microgravity environment.
And especially under partial gravity, because in '94 we did an experiment using a centrifuge microscope in space on the IML-2 mission, and we determined thresholds for sensitivity, and they were in the range for microorganisms. I myself did an experiment there, and plants in the range of 0.16 G. So it is very interesting that this, what we will expect on Moon or Mars, will be above threshold, so we can expect that biological system will use and experience the gradient still of acceleration there. And I don't think it's a problem to grow a plant on Mars or Moon.
Can I just add, thank you for bringing up the primary payload in Eucropis, that it's more than just a urine recycling. Of course, you know that the main objective is to grow tomato plants in different gravity regimes. So that ties in very nicely with some of the stuff that you're doing in Veggie. And yeah, there's certainly lots of data available on that. Yeah.
Couple of questions. Can you make bacon? No. So that's number one. I would be a vegetarian if it weren't for bacon, right? But here's a proposal for Lynn, Mike, and Anjan. What would it be like? Could we convince you to come back next year and have a taste test of some? Well, if you can figure out bacon between now and then, even better. But think about that and let's talk, because to me that also brings that experience that I now want to try finless fish. I never much liked, I'm too spastic to sit there and catch one, you know? It's like, no, I just want to eat one. But think on that, all right? So let's have a taste test in 2020 at H2M.
There are a couple of other companies that it would be interesting to make that challenge in the invite of, you know, giving these parameters, could you estimate what the mass would have been to produce this? And I think I spoke to you out in the hall beforehand. I know at least one other company that's working in the meat space using stem cells, Meatable, which I've had a lot of interaction with.
Yeah.
And yeah, my guess is they could go sort of the bacon route. You probably could go the bacon route. I did see on a vegetarian cupcake show that you can actually make very good imitation bacon with coconut husks and tamarind sauce, but that's another conversation.
And I'm so sorry, Matt, because one of the things I've heard multiple astronauts say that because of the pressure and all of the fluid that comes up from your lower extremities, taste becomes very interesting. So one of their favorite things is what shrimp cocktail or anything spicy. So I find it interesting with the plants you're growing that you are leaving room for a little bit of that spice. Is that, can we grow a hot pepper? Have you guys tried that?
We are working on hatch chili peppers right now for space. But yeah, no, the taste sensation, you know, they don't all say that it's muted, but most of them do say that it's kind of muted. It's a little like having a head cold because of the fluid shift. You know, your heart's still trying to pump the blood up from your feet, but gravity's not pulling it back down. And you'll see that a lot of the astronauts have slightly puffier faces in space. So they'll have a giant bottle of sriracha right on their dining room table. And, you know, so they do want strong flavors. And that's one of the important things that, you know, I think we need to think about for space food.
If I can add, I sort of gave a more general SynBio talk that these are perfect examples where synthetic biology can be involved so that you can get the flavor a little bit different, or maybe you grow a smaller plant so that you do focus more on the biomass that's edible, or you change the nutritional content, or you change the microbiome, or you change the ability to live it on, you know, these plants to live on urine or higher saline environments. So they're all, you know, or colder or warmer or whatever. We can do all sorts of things with the SynBio in conjunction with the food manufacturing to get you a better product.
Yeah, and I will say, just in direct response to Meatable, their CEO is a good friend of mine and an advisor to Finless. We used to work together, actually.
Okay,
but yeah, Don, awesome company. And so they, you know, there's a lot of these companies now, synthetic biology, like cell-based meat companies. There's about 40 that are funded worldwide now. We were like the third-ish, somewhere around there. Very few of us have any sort of eye towards space, but I think a lot of that is because there isn't really any incentive for us to do so. There are countries out there that are actually doing public funding of companies like ours. New Zealand has put forth, I think it's $300 million specifically towards projects like this because their economy is very, very largely based on animal agriculture, and they see if that sort of goes away, that they're in trouble. I know Israel's done a trade deal. China is sort of looking at this, but it would be really cool if America could put some sort of like incentive structure in place to like focus us towards space, because right now it's kind of hard because we're all very strapped for resources. But I would love to look into that as a project if we had like the worker power.
Well, let me put in a pitch for NIAC in that case. So talk to me later.
Yeah.
Hi, I'm Matt from the Planetary Society, and primarily for Mike and Anjan, but welcome other comments. Wondering what progress you're making on handling the structure of food that we expect, as in a fish fillet or whatever, and texture.
So there are several techniques that we use to recreate the texture. We use heat and cold quenching technology with a liquid nitrogen. So what we do is, you know, the 3D printing process works layer by layer. Let's say if you want to create something like fish, if you print first layer, and if you directly want to print the second layer, the bottom layer is not strong enough to hold the mechanical stresses of the second layer. And if you build subsequent layers, the whole thing will collapse. So what we do is that when we print the first layer, we have a blast of liquid nitrogen, and to the point where we don't want to damage the cells. So there is an equilibrium of how we can have good yield, but at the same time we can print on top. So we just need sufficient mechanical strength to build the structure. So after printing the first layer, we freeze it, we print a second layer, we freeze it, and then we build it up. And then we start incubating that structure. If it's a live cell structure, we start incubating that in the same chamber. So the way it works is we start heating up the bed and then we slowly move up. So we don't immediately, also, if something is very cold and you suddenly provide heat, it's going to go through a very stressful situation. So we slowly take it to the higher temperature and we start incubating. So that process has given us quite good control over texture of the food. And there are different techniques. This is 3D printing is more about process than the hardware. It's about, let's say if you want to print pizza, how do you create a texture of the crust so that it's really artisan pizza? Just to give you perspective, also the comment that someone made previously, when we started out, the taste was absolutely horrible.
But then so was a lot of frozen pizza, so.
It wasn't even, it was worse than that. Then
You're a salesman.
Well, so and then in the journey we met quite a few top chefs, and in fact they came to us, and it was all when we presented Human to Mars, it became viral. Our research work became viral, and chefs started contacting us that we want to help you out. So there was a top chef in New York City called Pasquale Cozzolino. It's a very famous restaurant by Union Station, and he gave us his recipe and he worked with us. He tuned our recipe, and after some time, after 6 months or so, the pizza came out probably as good as his pizza in New York. So having better process control gives us texture close to what we consume over here on Earth.
I would say that's much more detailed than what I'm about to say, but so there's a, this technology really comes in two pieces. The first part is to basically get cell mass that has the same like nutrition and taste, and from there you have like an ingredient that like can give actual nutrients to people and taste the way you want it to. And from there it needs to be structured. And so there's really two fields that you can delve into for this. One is food science. So this is sort of like the veggie burger type stuff that you're seeing, or like any sort of veggie steak, Gardein style stuff, Morningstar style stuff, say that 5 times fast. But basically that's in the realm of like food science, sensory science, texture science. And the other side of that would be more in the 3D organ printing side of things. And so 3D organ printing, if you see basically where that field has gone for medical applications, it is, you can see that the organs are being printed. It is sort of happening. The problem that they're having is to make these organs functional and to make them actually work. But for us, we don't really care if it works or not. We just need it to have the right texture. And so while they are delving deeper and deeper into definition and into structure, we're not sure that the human tongue can even really differentiate at that level. And so it really seems that for us, the problem that we need to solve is just to make it cheaper and faster. And so there's a lot of different methods of 3D organ printing or just 3D printing that can be used. There's like extrusion printing, as Anjan was saying, and there are faster varieties. Like I personally am super interested in stereolithography, which is sort of like printing 3D structures using a 2-photon laser. I'm not going to describe it because it takes forever, but there's videos online that would do a better job than me anyways. But yeah, I would say really, at first we're really focused on getting something that is a really great ingredient and then seeing how we can play around with that, which is around where we are right now. And to actually get something that is like workable, that is at a good cost point, will take some time.
Thank you.
First, I want to congratulate everyone. I think this has been fantastic. I've been sitting here buzzing, like, great job. So my name's Scott Bryson. I run a company called Orbital Farm, and where we are trying to pilot and develop agriculture projects for closed-loop agriculture systems. So we tie together with all of everyone here on the stage, where we combine biotechnology and aquaculture and hydroponics. And so, everything I'm hearing is just absolutely fantastic. Are you guys talking to each other? Is there collaboration together in the industry? From what I've seen, it feels like there's a lot of really amazing things at the different space agencies and companies, but no one's really coming together. Would a closed loop agriculture summit be interesting, or like collaboration together between everyone? Maybe.
H2M
Yes.
2020. Talk
Yeah, I mean, I think that it's a really good idea. I think there's been swim lanes, and, NASA obviously has a problem with swim lanes. And so we've had a workshop where we did actually have, companies that talked about some of the synthetic meats and other things as well. I think it's definitely a good idea, and there are so many people. I think it would probably be pretty widely interesting for a lot of people. So, yeah, we'll keep it in mind.
I know in our industry we have a fairly sometimes very collaborative point of view, and sometimes a very uncollaborative. But I think it depends very much on what conference you're at. If you really want to see us collaborate well, I would say there is a very unattended conference that you can go watch us do that at. It's in Maastricht in the Netherlands, and nobody seems to know that it exists, but there's tons of collaboration. Outside that, not so much.
Cool. Thanks very much again.
All right, again, I'm going to build on that question a little bit and take advantage of the fact that I'm the senior science advisor to the NASA Innovative Advanced Concepts program. And one of the things that we're very interested in is trying to get ideas like this and seed them into a potential future for NASA. And so, Gioia, I appreciated very much the fact that you introduced the concept of equivalent system mass, because what NIAC does is we don't fund you to develop your secret sauce. Okay, in this case, that actually applies. I often say that secret sauce doesn't really work, but today it does. But we don't fund you to develop your secret sauce. We fund you to tell NASA why your secret sauce should be of interest to NASA, and you do that by putting your idea or your innovation in a mission context. And so you're explicitly asked to consider such things that fold into equivalent system mass, so that if you make a really great fish burger, but you need to bring a nice oven along, and no, we don't have a nice oven in the station right now, but you're going to have to now add power for your oven. You have to consider how your idea fits into the bigger picture in order to be appropriate for a NIAC study. So NIAC is very interested in continuing the dialogue with all of you. Of course, you saw the NIAC logo several times on some of Lynn's charts. All we have to do now is just find a way to stamp NIAC on some of the subsequent speakers' ideas and anyone else in the room. And as senior science advisor, my role is to interact with you before you ever submit a proposal to make sure you're in the right ballpark. So if you're interested, just catch up with me sometime or find my thing. Not to distract from the purpose of this panel, which is to talk about the really cool stuff and how they're doing that.
I add to that, besides the fact I'm very grateful for all the NIAC funding, that as I was trying to imply at the end of my talk, is that often our concerns for getting something into space are very different than the concerns of the Earth-based people, where you might be worrying about your next quarterly report to your investors, or the petrochemical industry, or geopolitical concerns, none of which we currently have on the Moon or Mars. And so it's not always the same concerns going forward, and so then that relies on someone providing funding to apply it to the correct program, and that's not always the obvious thing either, is to actually then fund the secret sauce.
All right. We were actually told that there was an error in creating the schedule, and that we actually should have been ending at about 5, and so we somehow knew in putting this together that we should be aiming for 5, in spite of the fact that the schedule says 5:30. So I think this has been a very productive panel, and I've appreciated all the questions and all the attention, and I especially appreciate all the preparation that went into the talks coming in. So thank you all, panelists, and let's give them all one last round.